Hello students, today we discuss about the protoplast culture. The question on this topic is the given detailed account of techniques of isolation of protoplast from plant tissue. So first of all, what is protoplast? In protoplast is a plant cell without cell wall containing cytoplasmic mass bounded by the cell membrane. Means protoplast, it is the plant cells and it is without cell wall containing the cytoplasmic mass bounded by the cell membrane. Then cell wall is removed by the enzymatic process or mechanical process. In short, the protoplast is nothing but the cell organelles without the cell membrane. And the cell wall will be removed by the process of by the enzymatic process as well as by the mechanical process. So techniques or methods isolations of protoplast from the plant tissues are there are two methods of isolations of protoplast from plant tissues. Those are the mechanical process and enzymatic process. So first of all, we talk about the mechanical process of isolation of protoplast from the plant tissue. So you have to see in this picture. In that first figure is the plant cell. Then plant cell wall will be removed and the remaining part will be protoplast. So what is the procedure actually? So for removing the cell wall, get the plant cells from the peel of the leaf, then place the peel in hypertonic solutions. It occurs ex osmosis due to the plasmolysis shrinkage of protoplast take place. The plasmolyte cells are operates under microscope with microscalpel to remove the cell wall. Then thus the isolation of protoplast is carried out. So यहाँ पे क्या किया जाता है, जो लीफ का पील है वो पील लिया जाता है, फिर वो जो पील है वो हाइपरटोनिक सॉल्यूशन में रखा जाता है. जो सॉल्यूशन है हाइपरटोनिक सॉल्यूशन, they will be shrinkage the protoplast and then the plasmolyte cells will be operate under the microscope, and with the help of microscalpels we can remove these cell wall, and in this way the isolation of protoplast take place. Then what are the merits of this method? So it is quick method and it is less expensive. And demerits of these methods are it is skillful method. Often a result is failure. Then isolated protoplast cannot be stored or maintained. Only the skillful persons are removed these easily removed the cell wall. This is very often there is a very much chances for the failure. Also these protoplast stored or maintained is not possible. So these are some demerits of this mechanical protoplast method. Then second method is the enzymatic process of isolation of protoplast from the plant tissue. In enzymatic process, first one is a single enzymatic method. In that, first of all you have to take the leaf. This leaf will be sterilized, then it peeling off, then the leaf segment is plasmolysis, then it will be washing off, then it will be placed again in enzymatic incubations, then it will be filtration, centrifugation, and it will be stored in sucrose solution. Then this procedure of the incubation, filtration and centrifugation will be repeated 2 to 3 times. Then we can get the protoplast. So first of all we have to take the leaf. The leaf will be sterilized with the distilled water. Then again it is sterilized sterilization will be carried out by the 10% ethanol for 1 minute or 20% in potassium chromate for 10 minutes. Then peel off the epidermis of leaf. Then cut the mesophyll tissues into the fine segments. Carry out the plasmolysis of mesophyll tissues by treating with the hypertonic solutions. Then again this wash of the plasmolysed mesophyll tissues with sterile distilled water. Enzymatic incubation of plasmolysed tissue is carried out for 24 hours at 25 degrees centigrade. The enzyme mixture contains uh, macroenzymes, cellulase enzymes, mannitose, and S orbital pH 5.4. These carry out the filtration so as to separate leaf segment debris and protoplasm containing cellular mixture. Then again it will be centrifuged for the filtrate to get the cellulate of protoplast. Then repeat the process with enzymatic incubation to get maximum number of protoplast. Store the isolated protoplast in zero point 0.4 molar sucrose solution at 0 degrees centigrade. So in short, first of all, the peel of the leaf, take the leaf, it will be sterilized with the distilled water, then also sterilized with the chemical, then make the peeling of these leaves and make the leaf segments. Then it will be plasmolysed, then again it will be washed and it will be incubated in enzymatic solutions so that the debris or protoplast will be separated. Then these again it will be filtered and centrifuge so we can get the protoplast, and this protoplast will be stored in the sucrose solution. So in that, this is the single enzymatic process. In that also there is the double enzymatic process. In double enzymatic process, the same first part is the same, that is leaf. Take leaf, it will be sterilized, then peeling off, make its segments, then it will be plasmolysed. Plasmolysis will be takes place, then treated with the first enzyme, that is mixture of the macro macrozymate incubations in mannitol pH 5.8. Then use second enzyme, that is cellulose mannitol with pH of 5.5. Then again it will be filtered, centrifugate, and it will be stored in the sucrose solution. So these are the procedure for the separation of the protoplast in single enzymatic as well as double enzymatic process. So the next is the applications of